Hi, my name is Rosalie Elting. Today I'm going to be presenting How Low Can They Go? A discussion on compensatory feeding and floral community interplay in hummingbirds. I'm currently a lab manager at University of Washington, and this fall I'll be starting my PhD at the University of Montana. So why hummingbirds? Hummingbirds serve as an excellent model. They are a keystone pollinator and they drink a primarily liquid diet. And the plants they pollinate, because they're a keystone, they have a very tight relationship with one another. Hummingbirds also process all the water they consume through their kidneys, possibly limiting how much they can upregulate their consumption and creating a bottleneck in excretion. Hummingbirds will regularly choose higher molarity nectars at the cost of assimilation efficiency, suggesting they might do quite poorly when eating very dilute nectars. And lastly, they are regularly exposed to high desiccation risk with their large surface area to volume ratio, but despite that, the high water content of their diet means they have to work hard to remove all their water in their diet, and they are chronically diuretic with urine levels of concentration far less than osmotic concentrations. So all this led us to ask the question if hummingbird feeding rate is on a low quality nectar is physiologically constrained by how fast they can process water. And we hypothesized that since most water is processed through the kidneys, the hummingbird energy intake will be physiologically constrained when nectars are very dilute, and there will be a resulting maximum feeding rate where they can't upregulate any further. There's a cap in the rate that they can feed. So we researched this and answered these questions by going to 6 different sites in North and South America, representing a range in elevations and latitudes. We researched this question in 17 species, representing 8 different clades across 6 North American sites, and these species represent quite a range of body size as well. Our smallest species was about 3 grams, and our largest was 9.5 grams. The method for our work was used slightly differently across 2 different setups. All work involved a single trial for every nectar concentration. In our early work, we did them all sequentially and in decreasing concentrations, starting at 10% and going down to 2%. It was all done in a single day in the field. Our more recent work has taken these nectar concentrations and randomized them and done them often across several days. So not sequentially, and giving the bird 1 or 2 treatments a day. Both experiments involve live readings from scales or load cells to give us information about the consumption over time, as well as information about the bird mass. At first, this was a standard lab grade scale with a suspended feeder and perch, and now it involves Arduino load cells and coding to have live data from the perch and the feeder. So what do we expect? What do we expect our data to look like, right? So this graph is going to look pretty familiar through the rest of the talk. It represents nectar concentration on the x-axis, starting at 10 and decreasing down to 2%. And the y-axis represents consumption of nectar in grams of consumption per gram of bird per hour. So that treatment time, and then also adjusted for mass. And so what we expect to see is some increase in consumption as the nectar gets more dilute until it reaches a maximum, that being the maximum feeding rate. And at that maximum feeding rate could occur at one of many concentrations. It just depends on the bird. But once the bird reaches it, that would be a physiological cap upon which they can't go any further up. And then once they reach it, they might choose one of 2 methods we hypothesize. So either they would reach it, and then in method 1, they would plateau at that maximum rate, or method 2, they would peak and then decrease their consumption after that point. And this is what we saw. So this is 5 of the species represented here. The next graph I show will show all of the species. But in the left-hand side, you'll see both of those methods. We saw a drop promptly where maximum feeding rate is met at 4%, and then there seems to be a drop in consumption. We also saw the peak and plateau in the bottom left where the bird peaks at 4% and then roughly maintains that consumption rate. In the middle top, we also saw a plateau and drop. So they plateaued for a little bit and then eventually dropped their consumption. And that's sort of the same with the graph below it in the middle bottom. And then in the top right, there's something else, right? There's always an enigma. And this individual seemed to be doing something where it reached almost like a false maximum feeding rate at 8%, started to decrease and then really ramped it up at 2% again. And so we saw this in all the species. This graph has a lot of information on it, but the axes are the same. And you can see in each species, we still see this curve. So all these birds seem to represent a maximum feeding rate. They reach it at some percentage, usually 6 or 4%. And then after which point they either plateau or decrease their consumption. So then we ask the question next of, but what if something is going on here with their body size? So we looked at the body size impact on consumption, seeing if the bigger birds somehow handle these dilute nectars better. So on this graph, our x-axis is the bird mass, and our y-axis is the adjusted nectar consumption. And the dotted line here represents a nectar consumption of 0.5, which means that bird is consuming half of its body weight in the treatment time, which is 1 hour. And we can see that the lowest nectar concentrations at 2 and 4% on the bottom row are still where we're seeing birds eating the most, more than half of their body weight often, but it doesn't seem to have any linearity with body mass. So we don't see anything that suggests that the larger or smaller birds handle dilute nectars better. So then we asked, where does this situate these birds energetically, right? They're eating really dilute nectars and they're eating often a lot of it. So we represented this as sucrose acquisition. We looked at the grams of sucrose that a bird is getting per gram of body mass per hour at the decreasing nectar concentrations. And for all species, they're getting less and less sugar. When a bird uses that first method we outlined, where they reach a maximum feeding rate and then they plateau, the main reason they're getting less sugar is because they are being introduced to more and more dilute nectars and there's just less sucrose available. For the birds who reach a maximum feeding rate and then drop the amount of consumption that they are expressing, we're actually seeing a twofold decrease in their sucrose acquisition, because not only are they being exposed to more dilute nectars, they're also just eating less of it and they're getting less and less sugar. But what we also wanted to see was what would happen at concentrations higher than 10%. That's something that we haven't investigated, but this graph would sort of lead us to, because it seems like at some point they should be maintaining their sucrose composition, right? They should be upregulating the amount of feeding they can do as long as it's below their maximum feeding rate in order to maintain that amount of sucrose. That's not represented in this graph because we don't see much of a plateau, maybe some at 10 and 8%, but mostly they're just getting less and less sugar. And we don't really know what that level is. What is that percentage where they're able to maintain their sucrose composition? Another thing we asked resulting from this is, what if this is just a byproduct of dilute nectars, right? What if they really hate eating these dilute nectars and they will just generally downregulate how much they're eating as a result? So we extended this work at University of Washington. We did control measures where we would feed the bird 10%, so still a dilute, not ideal nectar for the bird, for 5 consecutive hour-long trials in a single day. So 5 trials, one after the other, the same nectar concentration. And we saw some natural variability. There was about a 0.3 gram range between the minimum and maximum consumption, but we didn't see nearly the same shape as we have in maximum feeding rate trials. So we extended that. We did that maximum feeding rate trial with this same individual, and we saw that there was a similar curve, sort of the peak and drop method it seems like this bird was utilizing. I've put a dotted line here to show both the control minimum and the control maximum to represent the range of variability that we saw within the control. And we could see that this bird is still using a very different method than it uses in the control, increasing its consumption until it reaches a maximum feeding rate, at which point it begins to decrease. We did this work in the field with Selasphorus rufus. We did a similar setup. The birds had randomized order of trials, but all within a single day for each individual. And we see a similar pattern. The maximum feeding rate is met at 4%. It has almost like a false peak like we had before, where it reaches sort of a peak at 8%, but then ultimately the maximum feeding rate appears to be reached at 4%. And we wanted with this to delve a little deeper, not only into how they're feeding, but also how they're behaving. So we looked into their activity budget, which I'll go into in a few more slides, And we also looked into the excretory byproducts, considering that this upregulated consumption seems to be very tightly linked with excretion. We began looking at urine. So we began using an oil trap below the perch in our setup, in which we collect any excreted urine the bird produces on the perch. And we began graphing this with nectar consumption on the x-axis, and then either the urine mass or the urine concentration on the y-axis. And our main takeaways is that nectar consumption can increase until over 2 grams, and for perspective, this is a 3.5 gram bird. But they can consume over 2 grams, at which point it seems like the amount of urine, the mass of urine they're producing is pretty stable. But after that point, it seems like they reach some sort of threshold, at which point they increase the amount of urine they're producing. The urine solute concentration seems to be independent of the nectar amount consumed. So it doesn't seem like as they consume more nectar, anything happens with the solute concentration of the urine. But we wanted to look into what the behavioral component is next. So what's happening beyond what they're producing and how they're physiologically behaving. So one of the huge benefits of having a load cell is we now can get some activity information, looking at when the bird is perching and doing anything besides perching. So getting some information about their active and their rest time. And it seems like in these 3 different individuals represented here, at each nectar concentration, we've assigned a proportion of time that they spend active versus resting. And it looks like the resting time is somewhat maintained as well as the active time until they reach some sort of threshold. So they're maintaining their activity until either usually between 6 and 4% or 4 and 2% nectars, at which point there's a big increase and a big peak in the amount of active time, which could mean that they would naturally be doing more foraging at this point or trying to find other resources that are more beneficial to them. So we next zoomed out a little bit to look at actual floral resource data beyond what we were using, which are very dilute, right? So we used some open source data from Tinoco et al. They did visitation work looking at hummingbird and plant species interactions. And then I created a graph that shows the sucrose composition of each plant species and plotted it compared to how much each hummingbird is visiting which species. So we have visit proportion compared to sucrose composition. And you can see quite a range of sucrose concentrations. However, none of them are as low as the dilute nectars we're looking at in this work. And we're seeing far more concentrated nectars in the natural floral landscape at this point. So this leads us to investigate the implications of this nectarivore relationship with the plants they're feeding on. Often flowers that hummingbirds feed from look like this. The plant is actively reducing the ability of the nectar to be diluted by rainwater. But this doesn't make a ton of sense. The hummingbird has an energetic need, and if the nectar is more dilute and they come to this flower many, many times, they're spending more time interacting with the flower's reproductive structures, potentially causing more pollination events. So it seems like it would make sense for the plants to create more dilute nectars. But these nectars also aren't just sugar. Refractometers are really good at measuring solutes in a solution. So we're getting percentages that we presume are mostly sugar because a lot of the times they are. But yeast and microbial communities in the nectar can change the composition of the nectar, where they are either consuming the sucrose and changing it into an alcohol or changing the solutes in it to create different smells or different structures. And it could be that hummingbirds have a preference for this, or that it impacts their energy acquisition when they're actually feeding at these flowers. We're also in a major climatic event where temperature, rainfall, and actual abundance of flowers could be changing, and that could have an implication for how much these birds get to the plants and what the plant's nectar composition is when they get there. So we kind of have a better pulse on what the physiological implications are of the feeding, and we see that there's a limit, but it's pretty plastic. They have a lot of upregulation before they get to a concentration of nectar where they start having a hard time getting their energy. But we're still looking into what this means ecologically for the plant-pollinator interaction. So in conclusion, with this work, we've shown that upregulation of compensatory feeding in hummingbirds does exist, and it has limitations, right? This maximum feeding rate is appearing to be limited by how much the birds can process their water. there's a huge behavioral component, changing the amount of time they're searching versus perching versus feeding. And once they reach this maximum feeding rate, whether they make a choice to maintain it or drop at that point. We're studying this maximum feeding rate at floral nectar concentrations that are not yet seen in nature that we know of, but well within the realm of possibility with floral shifts occurring. I'd like to acknowledge my funding sources, University of Washington, where I'm employed. I've also been employed through PIs who have grants from NSF and NASA, and was funded by the George Fox University Richter Scholar Program. I've spent time at Cedar Rock Preserve, Maki Pacuna, Santa Lucia, and Southwestern Research Station. And I want to thank a few people who have given me coffee, coding help, and allowed me to use their beautiful photos in my talk. I love talking about research, and I love also talking about outreach and getting more people represented in science. So if you'd like to email me or check out my website or follow me on Twitter, I'd love to stay connected.